Let's begin with a recap. There's a codon wheel. Certain triplets of nucleotides are used more often. Some amino acids have more than one codon, some have fewer. The next generation sequencing essentially proceeds. I did not quite bring that. It's still a recap for most of you. You take the DNA, you chop it up into pieces, you sequence the pieces, and then you find to assemble them, to realign them, to put them into the genome. This is much easier to do once you have a reference genome, as it is called, such as the one for human done in 2000. And it's much more difficult to do de novo sequencing for a new organism. That is behind the problem, for instance, for scooping just a piece of water and go through the ocean, or taking something out of the air or the soil, because then you get a lot of organisms that you have never seen before. That's why that is complicated. You assemble it all, you get a sequence. Today, Illumina is one of the leaders, at least for the shotgun short sequencing. These machines are called HiSeq. They're called HiSeq 10 because there are 10 of them. And the reason simply is that you have to buy 10 as a package. If you want to do sequencing in Germany, then all you get is one offer for 10. And each one of them is a little bit above $1 million. So 10 million investment for a machine. Now we're talking a level where typically universities cannot handle that alone anymore. Plus, there is a lot of output of data, and these machines have to be fed, and. So typically we have reached the level where, in fact, this has to be done on a level of governments or states or larger local regions, and that, in fact, is intended by Illumina. So the whole package, that's the business model. They have a capacity, as you see here, of over 18,000. This is on the level of human. And that is the price for 30 times sequencing of human. So we are below the $1,000 genome by now. This is what this implies. I talked about life. I talked about viruses that typically are not defined as life. And here's an exception, Mimivirus and many, many, many others. Somebody in the room noted this name here, which in fact I completely incorrectly traced to some possible German origin. It is actually Danish. And Tom Fenchel said this: we found a new species, and that was the Rønbjerg, the place in Denmark, after which it was called. And you somehow see the logic. So the conclusion from that, sometimes it pays off to look at what outliers mean, and sometimes it's sort of more or less boring, like in this case, but I still owe you that answer, boring or not. Back again, I said that before, Marco Punta contributed important to the slides, and Andrea Schaffhans. Today, I will simply go into what are amino acids, how are they strung together to form protein structures, what are bonds, energies, and what are domains, and possibly get into part of this. The reality and image part we already did. I just put it back in here because in giving you the PDF, it belongs here. So the main point is the message that whether that is the image of the dynamism of a soccer player or that is the image that you use, my argument is that both have different aspects of it. Both are not the truth, although this is a 2D image, it's a photograph of the truth, and we are used to believing that the photograph is the truth. But there's something in the truth that you miss in the 2D photograph, and that you possibly capture in interpreting this image, where that has an image of fire, has an image of energy. And it's the same thing on the level of the protein. So we have two different representations of a protein here. Here, in fact, you have different traces, so each line is one model. This is an NMR structure. So essentially what you see is different models at different sort of time points. The assumption being there are regions here where they are much more similar to each other, and regions where they are much more different. And the typical interpretation is more different means more movement. Okay? So in that sense, you immediately get an idea about dynamics from this image. This one here, that shows as the arrows the beta strands, does not have any dynamics in it. It shows a direction, that's the direction of the arrow here, and it shows how things fit together. Now, the in some sense, what you see here in the 2D, we're seeing 2D, but you see in a barrel, right? You don't see that here. So again, we're comparing visually 2D images, and they both tell different parts of the story. Okay? And this is what we have to keep in mind. So we're looking at 3D structure. The entire lecture will be about predicting 3D structure. But 3D structures are images. They are, yes, I measure for every single atom where in space they sit. But this is not what I'm after. What I'm after is how this thing interacts, how this object interacts with other objects. And in order to understand that, yes, the structure is the start, but it's not the end. And the structure is not true because even the image of an atom is debatable. And we'll get back to that in a moment. Constituents of proteins are the amino acids. You know, there's a backbone, there's a side chain. Isolated amino acid looks like that. It forms a dipeptide, di2. So they join together in the so-called peptide bond, where the backbones are linked in the peptide bond and certain parts are left out. Now when we look at essentially the simplest model here of two protons or two atoms, in this particular case hydrogen, two atoms being two protons coming next to each other, overlapping electrons, then one way in which we imagine this here is by showing the protons as balls, because they are spheres, and have some sort of rod between them. Okay? Another way of looking at it is this one here. What this one indicates, so you have much larger balls or much larger spheres. The important part here is the reality that captures is that you cannot bring these spheres closer to each other. Okay? So there's a limit. This may suggest, this bone-like thing may suggest that you could actually condense the bone and make the spheres come closer. This one suggests you cannot do that. Right? This is a reality you capture with this sphere model. While in terms of shape, the bone kind of model here appears to be more realistic. Right? So how do you compute shape? You compute shape by the size of a proton. That's the bone model. Or do you compute shape by the probability of two protons to be next to each other, and where you have a dispersion or a push away effect from electrostatics, which is more this model. In this model here, now we have to somehow get back to the bone model if we wanted to put the electron cloud. So all the possible places where the electron can circle on a larger sphere surrounding the smaller sphere. That would be sort of this model. And again, all of these capture different aspects of the story. You repeat the peptide, the dipeptide, in a tripeptide, in a pentapeptide, and you move on. Penta is 5. What is 4? Tetrapeptide. This way you join amino acids into proteins. In these proteins, again, the main line that you see is the backbone. The side chains stick out on both sides. The peptide bond, again, what you see is that when you join the whole molecule, there is a particular angle. So you have only certain angles at which we can move. You cannot have all possible angles. There are certain angles that are possible. And that essentially is the planarity of the peptide bond, as chemists would call it. Now, I call this rationalizing biophysical features of constituents because typically what we tend to say is we write down an energy function and we believe that this energy function describes the system. Rationalizing is my word to indicate that, again, what we are talking about are images, are models. This is not the truth. And ultimately, the way I can find out that it's not the truth is that if all of that were 100% the truth, then in principle I should be able to take these molecules and predict all its shapes. And I'm failing to do that. And part of the reason why I fail is that all of these are just images. So I'm not capturing the entire reality. That's why I call it rationalizing. Okay, let's get back into physical chemistry or some simple ideas. The covalent bond of the H2, again, I already spoke about that. The two protons are pushing each other away. Electron and proton are attracting each other. That creates an energy function, potential, that you can essentially simplify like this, where if you are very far away, you have an attractive force, and then if you come too close, you come to the point where the two protons push each other away. It's the simplest energy potential for the electrostatic of this example. Then we have a double bond. The double bond comes with another type of energy barrier for the rotation. That is behind the way in which you can rearrange all possible angles. That now constrains all the angles that are possible. Then we have a delocalization. This essentially is in this particular molecule, there are electrons that are somehow shared. There is a probability of shared electrons to be in some places, which is again called delocalization. That is sort of distributed. Polar bonds, this is about essentially you do not have charges, but you have polarity. So essentially you have a dipole moments. So you have somehow more positive charges on that side of the molecule than on that side of the molecule. It's not exactly positively charged. It's a positive potential versus a negative potential. And that creates a dipole moment. There's the ionic interaction. That really creates a Coulomb potential, just attraction. It's very simple. That now the repulsive, the attractive forms the potential I showed you before in a simple way is shown here. The strength is very, very different of different types of bonds. Just to remind you of that, they're non-covalent interactions, the hydrogen bonds. They are much weaker than covalent electron sharing interactions. So ion bonds are much stronger than hydrogen bond interactions, are much stronger than typical van der Waals interactions. So there's a large spectrum. This is a spectrum of a factor of 100 of all the forces that come together. But since you have way more of these guys than of the ionic bonds, they ultimately add up to making an important effect. Yes?
The percentage is better around
electron sharing is
the mass hand that smaller than this is larger than
this is much. Oh yes. I looked only at the amount. Thank you. I only looked at the number and I was very yes, absolutely true. This goes the other way around. So the numbers, to stick to that, the numbers are right. So again, salt bridges, ionic interactions, clear. In a protein, in fact not very often. There are pi interactions. Again, another example. So I'm not sure why I have the animation in here. So we have hydrogen bonds, we have ionic interactions, we have pi interactions, we have polar interactions. And they all are absolutely relevant for the formation of protein structures. Okay? Even the weaker ones play important roles, both for the way the structure looks and for the way it interacts with other molecules. So far, so good. Let's get back to proteins. Again, so we have different types of amino acids. Amino acids, the basic structure I explained, but now you can attach different features to them, and we get it to this in here. Arginine and lysine are two that I thought of to be positively charged. Again, this is a simplification. Depends on the conditions, depends on all kinds of situations, but they carry negative charges here. There are others that are polar, again. And what I missed here, most important, not sure why that slide is out, are those that are hydrophobic. Hydro, water, phobic, enemy of. So they want to avoid being next to water. Hydrophilic, philic is friend, so you want to be close to water. So essentially what you will see in a protein is that the hydrophobic residues will cluster together, and the hydrophilic ones will be on the outside. So if I took a protein of 100 residues, and so again, not again, let me just make that point. You have 20 different amino acids. Proteins are built up of these 20 different amino acids. When strung up in a protein, they're called residues. Okay? So protein-bound residues. Now, I only write down HN or something like that. Hydrophobic, not hydrophobic. So, do you believe I can fold the protein? Then now, actually, we now have to make this point clear with a model that's simple. So all a simple energy that says whenever you have hydrophobic residues, they want to make them close. Whenever you have hydrophilic residues, you want to put them on the surface. Okay? This is your entire game. Do you believe now in a computer you could simulate a protein? So CPU-wise, we can certainly do that. Do you believe we get somewhere? So we have a yes and we have a no.
Would work for some proteins.
Yeah, that would be great. No, so it has not worked for any protein. But now if we assumed that we can put the protein on a grid, which in fact proteins don't live on a grid, but if we assume we put a grid upon them, and a grid, so essentially what you do is you instead of enabling the protein to freely fold up in a open 3D space, you say there are particular positions, so you essentially make a grid with points, and you say that the C alpha can only occupy that point, that point, that point, that point. Okay, and you build up a sort of structure through the 3D space, and the move space is along these points that you allow. You space on a 2D grid. Obviously we have a 3D grid, but on a 2D grid it would be something like this, and you say that if you had a pair, it can only be connecting any one of these. Okay? So now if you want to energy minimize the simple energy that you just invented, you can easily try all the possibilities on this grid, and if you sort of projected a protein onto a grid, and it really doesn't look like a protein anymore, but it looks like something that if you allow yourself a grid, then we can predict the protein of 100 residues. Now, it takes sort of some imagination to call that protein that I took onto the grid that protein. It certainly is not the protein that we have, and it certainly cannot, it does not do a very good job at predicting dynamic behavior of the real protein. But
I didn't mean like dynamic behavior, but for example, some structures could be, some known structures could be reconstructed using a special type of lattice.
Yeah, that is the lattice, right?
Yeah.
So again, but this is. Oh, we have to make the movie, the photo. Could you guys, but we don't need you for, yes we do need you for that. No, we don't need you for that.
What's going on?
Yeah, I don't know. I'm just a poor teacher, why would I know anything?
Do you want me to get a camera somewhere?
Then I will not forget it. Yes, because I forgot it on Tuesday. Thank you. It's in the, I'm not going to say that on camera. It's in my room in the lower drawer on the right. Okay. I'm not going to say that on the camera. My office is open, of course, and anybody can get it. Anyway, so getting back to that point of the lattice or the grid, you take a real protein, trypsin, for instance, a very short one, and you put it onto a lattice, a 3D grid. Then what you get is the closest way of matching the 3D structure to that grid, but it's no longer that protein. So now if you're happy that this is in fact that protein on a grid, which I happen to be not happy with, but if you were happy, then you're right. So then we can actually fold the protein. It's true. Now the next issue is that these, in some cases, the algorithm will find different alternative solutions. And now, is one of them contradicting? The next question would then be, is one of them contradicting the real protein? And the answer to that question is, since the grid is so different from the real protein, no. Mostly compatible with it. So then the story gets complicated. So science is a lot that you have to, please remember that. When you come up with an assumption about something, you have to find a way to prove yourself wrong. So if everything you find is right, then this is religion. You always knew the answer. Science begins if you define a model or a way in which you can prove to be wrong. Right? And this is a little bit the complication with I mean, again, it's not because it's not scientific, but the complication is that the grid is just too far away from the actual reality of the protein to be sure that we really got somewhere. But nevertheless, let's get back to hydrophobic. That is in principle on this slide, but not visible, because I somehow took the wrong slide, or the marking of the hydrophobic residues is missing. But we do, by the simple model, they're hydrophobic and hydrophilic residues, we understand a lot of what we see in terms of protein structures. So one thing is absolutely clear, even if I cannot possibly simulate from principles of molecular dynamics the actual proteins with these two simple forces, they are the dominant explanation of what we see. And they carry us very far. It's not enough to really fold proteins. But it is enough to understand or to assume or rationalize, and then we're back to the word I used here, to rationalize what we see. It's not predictive. It's only to make ourselves feel happy that we understand why what we see is what we see. They're macroscopic substructures. So again, let's begin with the simplest thing, protein sequence. The potassium channel looks like this. Anybody in the room, is that a typical protein? What can you say about this? Can I have some observations about this protein sequence? Whatever it is, whatever you, with a wealth of information you come to this lecture with, wherever it comes from. What comes to your mind when you see that? When you see this slide, everything that is black on this slide, everything that is black between here and there. So below the word protein sequence and above the bottom here. Anybody can observe anything that's unique or not unique, or again, try to learn to observe data you find. Let's call that data you find. It's not, but can I have some observations?
I think it's fairly long.
Fairly long, right on.
80
Next.
amino acids per line. If it's 80 amino acids per line, then it's around.
That's long.
Yeah.
It's certainly not an average protein. Yes, correct. Next observation. And that, in fact, was the first I was hunting for.
It's like for function, actually quite normal. It's ATPase. I mean, for ATPase, actually quite normal for me.
That is very true what you say, and that obviously is already sort of more detailed knowledge, more background knowledge. Is a membrane protein is another piece that you would see, or membrane-associated protein, and it's an important sensor for the environment. So you will in fact see, in this particular case, a lot of charges, and you will see an overrepresentation of glycines. Okay, protein structure. What you very often, yes?
We had a question.
No, it was no question. It was like A and R's, not A and R's, but
There's lots of A and R's, so yes. So you get thanks, Dimitri. You get mobility, and you get in fact positive charge. So that is in fact, in this particular case, you spotted that very well, is related to the function. You may, in many Wikipedia sites and many books, you may read this utterly nonsense historical connotation of primary, secondary, tertiary, quaternary structure, and historically it is absolute nonsense. So there was a historical reason. Today that has no reason for being other than whatever. This sort of was thought to be the first thing you sort of see. It doesn't have a structure, it's just a sequence. It has nothing to do with the word structure. Secondary structure is sort of a substructure, is sort of the second element, and in fact it's also not true, because the first thing actually people saw was this one. The three-dimensional structure, and there the word tertiary comes from three-dimensional, but it's sort of a misnomer for 3D too. Quaternary, four-dimensional structure is also pretty nonsensically. The way that originally was explained is by, this explains how proteins interact. Proteins interact, or units of a single protein interact. But this is actually also more complicated, why this is nonsense. And now I made a big problem, making a big mistake here. I should have put what I consider the reasonable explanation, but we get to that in a minute, or the reasonable way of calling things. One thing that is very important are secondary structures. So this is one that you see here. Those are the hydrolyl angles that essentially come from the peptide bond, so you can turn only in a certain way. And this one here indicates one turn of a helix. One turn of a helix is essentially from here to here. And I and I plus 4, the residue I and the residue I plus 4, they are bonded by hydrogen bonds. So the next one, the next one, the next one. So essentially you build up a helix with a lot of hydrogen bonds. Why do we have secondary structure? Okay, we get back to that in a minute. Here's another secondary structure, the beta strand. So now again we have a little bit a set of different words used for that. The hydrogen bond now is I and I plus 2 hydrogen bonds, but now the hydrogen bond to something further away. The notation is typically, this is a strand. When strands come together, they form a sheet. A single strand doesn't make sense. Essentially, the hydrogen bonds are only formed if you pair up two strands into a sheet. The idea of a sheet, and this is what you saw often, or what you will see again and again as arrows, where the direction shows from N to C, from the beginning of the protein to the end of the protein. So the arrow here sort of shows the planarity of this peptide bond background, and you see that the planarity is such that they are aligned in terms of local 3D configuration, right? And that's another reason for drawing these arrows. So these arrows, in fact, convene more than a direction, and the fact that these are strands, they also convene something about the local bonding pattern or arrangement. Can you spot the secondary structure in here? So let me make this clear before. Blue and red here are not secondary structure, I believe. Are they? Well, it's difficult to spot. Can you spot secondary structure here?
Analysis, right? There's Alice up there.
There's a helix?
Okay.
Images and reality. So this is more reality in some sense than this. But secondary structure is an important reality, and clearly you begin to see that here and not in the other one. But you see more, right? Anybody sees anything other than that helix? Can you spot another element? Somebody else? No? You don't see anything else? Really? This is sort of a turn of a helix. This is sort of a turn of a helix. This is something, no? But something else, not helix. You don't see anything else that is not helix? Okay, when there is no arrow, beta strands are sort of seen, or sheets are seen by suddenly many lines are parallel. So this is a sheet, and here's the coloring. In fact, I overdid it with that one. Again, they are stabilized by hydrogen bond formation. Let's get back first into the angle story and begin with the first slide in the direction of shapers and shakers, Ramachandran. Ramachandran, born October 22, lived until 2001, was an electrical engineer from physics, Raman spectroscopy, that's where he did his master in optics, went to the Cavendish lab in Cambridge, did crystallography. That, in fact, is the first crystal structure that came from the Cavendish lab in Cambridge. Went back to India, shifted to biology molecules, and for instance, was the first to show the triple helical structure of collagen. In '63, he published a JMB, Journal of Molecular Biology paper, and the analysis of big bone conformations. Those are what went into history as the Ramachandran plots, and they essentially show that when you look at all the dihedral angles here, these two angles, phi and psi, psi and phi, phi and psi, then, and you plot for a set of proteins, in this case that was that set of proteins, you plot all possible occupations of every pair of C alphas, so the main backbone atoms, then you see this distribution of angles. And in this distribution of angles you see this is alpha helical and beta strand. So you essentially see regions that are not allowed at all. If you look at that today for the entire protein database, you will find, most likely, I haven't looked at it since 20 years, but most likely you will find points everywhere due to the variety of different reasons. But if you now made this again as a mountain, so where the height is proportional to the number you find, then most likely you would see that, yes, there are some points, but essentially the mountain would look exactly like, very, very, very similar to that. There are some exceptions, but essentially this is still the original plot. Again, so there are different ways of plotting, showing proteins, cartooning them. Here is a disulfide bridge, cysteine and cysteine form disulfide bridges. Okay, let's get into a different level, the level of domains. What are domains? Domains are units of proteins. What are they? Ideally, somebody who did not take last semester's course, because I believe we talked about domains, didn't we? No, we didn't? Then you're allowed to answer.
Some part of proteins are repeating in other proteins.
So a domain is a region that you find again in another protein. Is that the definition of a domain? Okay, so AR. So she saw in the potassium channel, she saw a lot of ARs. Is that a domain? Well, so in this particular case, okay, we saw it repeat in the same sequence, but I bet my life that you find AR in many proteins. So is it a domain? Somebody. So he ventured to come up with a definition of a domain. Are you happy with that definition? I mean, he did not say AR. Let me just observe. I'm twisting your arm because I enjoy doing that. But now, in this twisted version, AR is a domain. Are you happy with that? No, why not?
Usually domains are associated to some function.
Okay, so domains are repetitive units that are associated to some function. Okay, so oh God, I'm blanking. I'm really blanking at the moment, but K H K D I. This is totally invented. But most likely, this is associated with a particular function. Is that a domain? Again, okay, I now my head is here. This one, I know the function of that one. I can tell you it's found in many, many, many proteins in humans. Is it a domain?
Should we have kind of evolutionary background to define whether something is domain? Because
So evolutionary background, what do you mean by that?
I mean, like maybe there's some kind of like domain. The way I read it is like because it came to reality through, let's say, replacement of like relocation of gene into other gene, right? And a combination of domains.
Okay. I will continue on the discussion of the domain, but let me just follow up on that last point, because it just happens to fit onto the next slide. Again, I'm twisting your arm slightly, but in the slightly twisted version it fits. Looking at the gene from the beginning to the end, we have exons and introns. Introns are spliced out, as it said, so taken out. Only the exons in this particular case here, 3, move on to become the final protein made up in this particular case of 3 exons. Those 3 exons are the protein. Are these 3 exons the 3 domains of that protein? In fact, this has been claimed very, very often in the literature, and from the 60s, Walter Fitch was one of the first, when he discovered that structure in the gene, suspected that this may be related to protein domains. It was too early. At that point, domains were not really seen, so there was no way for him, when he had this idea, to actually check whether it's true or not, because there was just not enough 3D structure available for proteins. Later people checked. And in fact, there are still publications and claims in textbook about this today. Is that what you mean by evolutionary related? These introns. So in intron, you can, an exon. You can take an exon and move it to another organism, and we do in fact see that. We do see exchange on the level of unit exon. Then, given your statement, you would state that these, in fact, are domains, right? Anybody doubts that, or has an issue with that, or wants to say something about it?
Okay, it does not always,
correct. It's not always the case.
yes, because I don't know,
Oh, yeah?
associate like structural subunits with domains. This is also not always the case, but it depends on your definition of domain.
Yes.
So you could look into SCOP and they have domains, or
yes, but you're getting a little bit ahead of my lecture. I never said nothing about SCOP. SCOP has a model of domain that is very similar to what you just said. Actually, it uses the word evolution and it uses the word structure. So somehow both of your words come in the head of Alexei Morzin, who did SCOP. But ultimately what you say is domains are related to some structural unit. So one way of putting that is the idea that I look at the three-dimensional structure and I see something that stands out, possibly I see again and again in evolution, and as a repeated theme, as a repeated three-dimensional unit, and that's what I call domain. And that clearly is one way of looking at it historically. That's somehow what guides SCOP. Another way of looking at it that is very, very related is just an additional step. So you take a protein sequence, you throw it into solvent, and it adopts a unique three-dimensional structure without any partner being involved. So you do not, in that soup, need any other protein, sometimes you need metals, but in principle, that's the information for how the three-dimensional structure is formed is contained in that sequence. Now, if you took a subset of that sequence, you threw it into that solvent, do I have an example for protein? Yes, we can easily imagine that we take, for instance, we cut out this part here, or we cut even the entire helix, and we throw it into solvent. Most likely it will not adopt the unique three-dimensional structure. Most likely it will not fold right, because it's somehow incomplete. And one way of looking at a domain is to say this is somehow complete. In terms of 3D structure, complete means surround, means that the contacts inside are all full. While this one here would just, you have a helix that needs to be packed to a beta strand to another helix because they're hydrophobic residues. You put it into solvent, they are exposed to water. They will not like that. And that will not create a happy being, happy helix. So the other idea really that is in fact the same idea, it's just another way of formulating that, is I look at as domains, I consider units that fold independently. In some sense. That also actually helps you to understand how big proteins can fold very quickly. You would see there's some rapid collapse on the level of a domain, two domains, and then they sort of come together. And that in fact is one of the dominant ideas how folding happens. Now this whole story that I'm talking about is a usage of the word domain that is highly biased by structural biology. Most of the time that you read the word domain in PubMed, people refer to it as something that is in fact KDEL. There are publications that say KDEL is a domain because it's a functional motif in some sense. So twisting your arm is how many experimental biologists, even computational biologists, see the word domain. Sometimes motif, KDEL, you could call it motif, short for peptide, tetramer, is in fact a tetrapeptide. It could be called a domain, and people do that. That is not the structural idea. Now, if I use the idea that you had as a structural idea, and for the course of this lecture, this is the way our guiding principle is: a domain is sort of a self-contained unit that indeed evolves separately, that indeed has a similar function somehow, has to do with the function, but also folds independently. In this idea, introns do not map to domains. Yes, there are cases where they do, but this is not more than would have been expected by chance. Most of the time, this is entirely wrong. Okay? There's still publications from 2015 where people claim that this is true. It's just one of those things. Here's another story. We have a story where we have one organism, P, that has a protein A, and a protein B. There's another organism, P2, in which you find protein C that looks like that. So those of you who have not been last semester, why could that be the case? Yeah?
Maybe a protein with two different functions.
So
they don't use two proteins for a certain reaction, just they put the two functions for protein A and B into one, and so it can work in two ways.
I'm sorry, I'm not quite getting that. So you're saying that A and B actually have a similar function?
No, but they can be fusioned into one protein with both functions from both proteins.
Yes, but why would you do that?
Evolutionary reason?
But what do you mean?
Maybe it would be energetically more efficient if they do that. Maybe
What could it be more efficient for?
Maybe both are interacting normally.
This is a typical explanation, but why would it be, in some sense, let's get back to his point, why would it be an advantage to have them close if they were interacting?
Then they're always expressed in the same level.
That's the point. So in that sense, energy in some sense would be defined as probability of coming close. And again, so the word energy for process in the cell, I'm a physicist, and the way I at some point in my past I was a physicist, and the way I think about energy in biology is not really, it turns out to be very misleading. But the probability of making mistakes, so being close together when you need to be close together, that really may be an important criterion for biology. You're readily available at the right moment. And if you want to interact, that may be an advantage. It may also be, to follow up on your idea about different functions. It may also be true that they do not themselves interact directly, but have to be part of the same pathway. And so both of them have to be there for doing something, okay? And again, in the same sense, since they are co-expressed because there's one protein now in this organism, you increase the probability that they would do something like that. So, yes. Now, in some sense, that implies that you can, by looking at sequences, possibly get out something about domains. Let me again show you an example where you see in 3D a domain, a unit that is repeated in a different unit on a slightly different scale. And here, the slide is from Christina Rengo. Again, you have a protein here, a protein. You have another one much larger. In this much larger one, you see a repetition of the small region of the motif, the domain-like structure. On this slide, she gets to the next level, describing with symbols subregions, domains. Now, that is one domain. In this particular case, is slightly overstating the case, but in this particular case, you would have 6 domains and many more. And what you see here is that you have the repetition of the same subunit. In some sense, coordinated. So all of these proteins, for instance, have a circle here. Have this domain here. Somehow in the middle they have this yellow triangular domain, and then most of them have another circle here. And that is the kind of theme by which you build up functional motifs, and by which you sort of explain functions through the aggregation, the usage of substructures. Substructure then being this triangle, being this theme of beta strands packing together against helices. Okay? Okay, if I showed you an alignment like that, and what I mean is that protein A begins here, ends here. Protein B, begin, end, has in this region a sequence similarity to A. So wherever these bars, these lines overlap, I imply that they are similar in sequence, and so forth and so on. So A has a region where it is no similarity to any of those, has a region where it is similar to B, C, and F. What, with respect to domains, do you see?
There are several potential domains, right, among these sequences.
Yes.
Like, for example, the B and C are basically the same protein with some different suffixes, and D and E, I mean, basically, D is a domain and which is containing E and F, right?
So you would argue that what you see is that the domain is from here to here.
Around that place, and then the C should be also a domain because it repeats in I and B, right?
The C repeats in
B and A. You see that.
So, but what is the domain? Where do you see the domain?
Around the C and like the.
Can somebody else? So now let's assume that we said that there is one domain that is somewhere in this region here. Okay? And I want you all to tell me when the domain begins. Okay, fair enough. Where does it end?
Maybe there or a bit later.
Yeah, somewhere there, right? Well, okay, you get entirely the idea. And this is exactly, plus minus, but you have to somehow cut it such that there's no overlap, and then you essentially see the domains. Unfortunately, this is the best way, and I will come back to that, and this is the best way in which we, from sequence alone, can predict domains. Unfortunately, life is not that simple. The hope had been for many people who try to predict domains from alignments that reality is like this. But the more sequences we had, we found that reality was much more like this. So meaning that in this image, what I imply by that is, okay, where it ends plus minus a couple is not a difference. Your will is just a matter of a threshold, and it doesn't really matter where exactly you put it. Do you put it here? Do you put it here? It's not defined by the problem, but it also doesn't matter. In this image here, if you take all of these seriously, you immediately see there's absolutely no domain. It is not a matter of a threshold. No matter what threshold you use, you will not come up with a reasonable classification unless you throw out some of the data. But if you take the entire set of data, this is hopeless. And reality is much more similar to this than to that, is unfortunately the reality. What I'm going to show you now is a bunch of databases. SCOP, somebody said that word. SCOP is one of the databases that classifies proteins from 3D structures into domain-like regions. And what I'm showing here is the average length of a domain and the cumulative percentage of all the domains in a database, families. So they're grouped by sequence similarity. And SCOP in this figure is the only one that really uses 3D information to define domains. So really follows up on this image that you had where we have a sort of compact 3D object, right? You look at the 3D structure, you identify the domains. And what you see is, let me just pick a point, 50% of SCOP has a length shorter than 120, okay? So some of them are very short, but very few are very short. Somehow, I don't know where you put the middle here. Many of the ones of the first, the smallest 50%, many of them are somewhere here, sort of in the 70. And then there are very, very, very few. So the remainder here, we are reaching 95. The remainder is domains that are longer than 400. So almost all the domains in SCOP then are shorter than 400, okay? Now, let me pick out one particular example that you may be aware of. PFAM is the attempt to classify proteins into families using sequence alignments without using structures. So essentially, PFAM is a much refined way of what we did before. You look at these alignments and you identify domain boundaries. It's refined because they do it iteratively. They do it driven by a lot of expertise about what the protein does. What you observe is that blue is below black, meaning that the average PFAM is much shorter than a structural. The average PFAM entry is much shorter than a structural domain. Okay, and then we go, here's another similar approach that is much lower than that.
Should be much longer.
The other way around. Yes, thank you. It's much longer. Why is PFAM much longer? There is some problem in this. This cannot be true. There's something totally wrong. I have not shown that slide. There's a paper I have to dig out, so I'm going to look up this paper again. Maybe we even published the wrong. This cannot be true. Absolutely not be true. I'll get back to that. Some common assumptions and facts about proteins. Longer proteins are rarer, less often observed than short ones, and prokaryotic proteins are shorter than eukaryotic. So yeah, longer proteins are less often observed, very long ones at least. Are prokaryotic proteins shorter than eukaryotic proteins? Anybody says yes? Do you never read books or papers? So the vast majority of publications claims yes. I don't understand why you're right, but you are. They're not. What I show here is the number of residues in a protein, and essentially eukaryote, prokaryote, and archaea. Now prokaryote is green, archaea is red. And you see that essentially the distributions, the main part of the distribution, is the same. Most proteins are similar between eukaryotes and prokaryotes. There's an exception to that, and that is on the tail here. Proteins longer than 500, the point here is 1,000. So while over 7% of eukaryotes are longer than 1,000, this number is only 1%. for prokaryotes. So that really is an important difference. The difference in the tail, not in most of the distribution. Okay. Let's get to the next ones. Some proteins are multi-domain, most are not. Don't look at me like a question mark. I'm asking the question to you.
Can I say no?
No. So most proteins are multi-domain is what you're saying?
I mean, most proteins that we learn about are multi-domain, but I don't know about proteins that we don't learn. I mean, maybe it's because bias and important proteins are multi-domains, not
Sure. Okay. We'll get back to that. So I believe the room is completely behind. Most proteins are multi-domain. Most multi-domain proteins have more than 3 domains. I'm not talking about, oh, I didn't show you the ADH yet. I'm not talking about the identical copy of the same, so dimers. I'm talking really about domains. So most proteins have more than 3 domains. Anybody says yes? Yes. Anybody says no? No. Everybody says yes to that one. There are more proteins with 3 domains than with 1 domain. If we follow up on your model, or we learn about is multi-domain proteins, then the answer would clearly be yes. Okay. Short insert here into chopping proteins into domains. And essentially it's the same thing that I showed before. So if you wanted to identify domains from sequence, the best thing you can do is you look at 3-dimensional structures in the scope way, and you look at regions that are densely packed. Call that a domain. So say we start with this protein here, and if we found PDB hits to PDB domain, we cut that out. Then we, in this particular case, have a short overhang of just 10, 20 residues. We do not call that a domain because domains are longer than that. We cut it out. Okay, at that point, cut 2, we remain with this one. Assume we find a hit in PFAM. In fact, PFAM domains are much short. PFAM regions are much shorter than structural domains. Whatever went wrong in my previous slide, I can't currently explain. That's the next cut, but it's the next best thing. If we want to define a domain and we don't have a 3D structure, then a PFAM is the next best thing we can have. So we cut again. Now, in this particular case here, the remaining fragment is long enough so that I will keep going with it. So is this one in my model. Now assume in the SwissProt database, what I find is a bunch of proteins. I don't know whether you see the light blue. So I mean, there's blue, blue, and there's another blue here, and there's one along these lines. Essentially, that's the model that I said biology is not as simple as that. But if we found an example where biology is simple as that, we cut. Then we have a region where we don't find anything. We don't find PFAM, we don't find an example like that. And in this particular case, then we would today say that the protein has 5 domains. And I'm not entirely sure why I do that as an animation now. I'm sorry for that. What this one now answers is what fraction, if I did that to a large data set, including all human proteins, then as a percentage of fragments and as a percentage of residues. As a percentage of fragments, so fragments is essentially these domains. I call them fragments because for some I know their domains, for some I don't. So as a percentage of the final fragments that are created, about 20% go by the PDB. Another 27% go by PFAM. About 4% go by this alignment works kind of cut. So you see a clear pattern in the alignment. And then there's some fraction here where it looks like it's a full domain. So they are short enough to be a domain. And that is the part where it could be a domain, a very long domain. It could be that the cut remains to be done. But this is sort of the ballpark of what goes into my next slide. And I tell everybody never use animations. Okay. Number of domains, percentage in proteins, green, protein database, proteins of known three-dimensional structure, where you clearly see 60%, the majority of all the proteins, are single domain. Whatever you see in your courses, if you look at three-dimensional structures where we can define domains, most of them are single domain. If we take this algorithm that I showed you before, and we look at things for which we do not have structures, include a much larger data set, we see that PDB is absolutely not representative. But in fact, the blue ones here, now the single domain is in fact less often observed than two domains, and clearly, so single domain is less than 30%, so most proteins are multi-domain proteins. Okay? And that is, the blue is much closer to reality than the green. In fact, the blue still underestimates, because here we have a bunch of things we could just not cut anymore. There may be 150 residues long, we didn't dare to cut, but maybe one day I'm going to see that there's a cut. Okay? Now, so that answers all of these proteins, these issues. What I show here is number of fragments, I call them fragments, pseudodomains in a protein. And I ask, what's the average length for PDB, for proteins of known structure, so by subset, that in fact has to be shorter because people cut away in order to do the structure, everything that is sort of not forming a structure. These two sets are real full-length proteins. This is essentially dominated by prokaryotes, and this is eukaryotes only. You see that, yes, there's some sort of tendency that eukaryotes are a little bit longer here, but these two curves are completely parallel, meaning that so for proteins with one domain, eukaryotes are a little bit larger than prokaryotes, but for all the subsequent domains, they're actually the same. And all the subsequent domains, essentially, so the slope here is you rise by about 100. So an average domain is about 100 residues long. That's as simple as that. Okay? So now we have an idea. Domains typically are about 100 residues long. And the vast majority of proteins are the ones that you have always seen that have multiple domains. For structural biologists, that is unusual and unexpected because most of the ones in the PDB have only one. Prokaryotes do not have more domains, fewer domains than eukaryotes. Only for this part here, for the ones with very, very, very many, that is again this slide. Oops, this slide. Slide I showed you here, that the very, very, very long proteins are more prevalent in eukaryotes, and that's why eukaryotes also have proteins with very many domains. Okay, now we get to the issue of clustering. So there's a function space, there's a structure space, there, in this particular case could be a sequence space or an experimental feasibility in this graph, and we want to cluster proteins. We want to cluster proteins, for instance, if I imagine that this is the universe of all the proteins, that an island is a structure. So all the proteins that adopt a particular three-dimensional structure. Some islands are green, I know the structure. Some islands are white, I don't know the structure. How much of this clustering is in the method? How much is in generic comparison method? So my point is, essentially what you start with, in some way, are points. And the moment you put line around these points, the moment you group them into a circle, into this figure, that moment you imply biology. That moment you imply classification. But the points themselves are not classified somehow. You can, now, this is an important point, because if you wanted to classify them by function, maybe the functional shape around them would be larger, smaller, even a different shape than when you classify them by structure. Those of you who attended last semester's lecture, so to predict protein interactions or predict enzymatic activity, it is actually a much smaller circle for these two different problems. So protein-protein interaction is a much smaller circle than enzymatic activity, is a much smaller circle than localization. So different aspects would give different circles, and in fact also different shapes. So the circle is not always the right shape for that. But importantly, the grouping really is biology. And that is, in fact, one of the big challenges for grouping proteins. Recap, cells are densely packed, surrounded by membranes. Proteins come at very, very, very different scales. When we look at proteins, we visualize them in terms of their shape. This is a slide from Natasha Wood from Cape Town, where she looks actually at T-cell surface receptors acting or responding to the glycoprotein 120, put down to sort of more schematic drawing where you have the viral membrane. You see that, and it's highlighted on the next one here, there's a particular loop that is very relevant for the way the GP120 enters the cell, the so-called V3 loop. And in fact, a lot of the targeting for drugs is on this loop. This loop, in fact, looks like that and has different potential to link glycan sugars, and that is exactly what people try to attack, to change the function to effect develop a drug. There are different protein secondary structures. I did not finish my statement. Why do we have secondary structure? Essentially, you create by hydrogen bond formation a local region of that is highly energetically favorable. So forming a helix helps you to bury energy in the molecule, essentially, and the same is true for the beta strand. We will talk about other possible secondary structures and about how we can learn from secondary structures to compare proteins, which in fact is what we will do on Tuesday, 3D comparisons, and then toward the end of the week, on Thursday most likely, we'll begin with alignments, and then begin with comparative modeling and get into secondary structure prediction. Thanks for your attention.